This tutorial looks at the manufacture of pharmaceutical drugs and compares batch and continuous methods of production, along with looking at the safety of drugs, looking at the manufacture of the materials going into drugs, and why drugs have to be pure. I'll look at why batch processes are often used in the production of pharmaceutical drugs, whereas continuous processes are usually used for large-scale industrial chemicals like ammonia, and then the advantages and disadvantages of each of these processes. Many pharmaceuticals are only needed in small quantities, for example those which are used for cancer cure. Therefore, it's too expensive to set up an enormous factory to manufacture them. They're made in small labs such as this. This batch process uses small-scale equipment which can be cleaned out and reused for making other similar drugs. So these kinds of pharmaceutical drugs are only made when they're on order and often will only keep for a short amount of time anyway. And they need specialist workers to make them who are usually very highly paid and well trained. Although the majority of pharmaceuticals are made by batch processes, these one-off processes if you like, there are some which are made in continuous production. For example, things like ibuprofen, aspirin, paracetamols, Ventolin for asthmatics, these sort of chemicals which are needed in large quantities. It would be worth creating an enormous factory to manufacture them. Continuous processes would go 24/7, and although the equipment would be very expensive to set up, it works out cheaper in the long run because the whole process can be fully automated and requires very few workers at all. Summarising the advantage and disadvantage of each process, the continuous process is better for large-scale production, as I said, for industrial chemicals, but also for things like aspirin and other painkillers, whereas the batch process is better for small-scale production, for example, of anti-cancer drugs. Whereas the continuous process keeps going 24 hours and is low maintenance, largely automated and computerized, the batch process is a one-off process. It requires a high amount of maintenance and is labour intensive because the equipment needs to be monitored, emptied and cleaned and refilled each time, whereas the continuous process doesn't require emptying and cleaning, it just keeps going 24 hours. Now, the disadvantage, I suppose, of the continuous process is it's got very high setting up costs. But because the same drug is manufactured day in, day out, there's a low cost per unit. Whereas the batch process has got much lower setting up costs. A normal factory can be used, or a lab, but it does create a high cost per unit because each of the drugs are manufactured in small quantities and by highly trained and highly paid workers. Here's a past paper question. Stowmarket Synthetics own a chemical factory. They want to make hydrogen peroxide. It could be made by two methods. Look at the table and it gives some information about the two methods used to make hydrogen peroxide. And then it says Stowmarket Synthetics decide to make hydrogen peroxide by method 2. It's because it's cheaper. The use of a catalyst helps to make method 2 cheaper. You can see that a catalyst is needed for 2 but not for 1. Okay, and then it says the use of a catalyst helps to make it cheaper. Explain why. Well, a catalyst will speed up the reaction. So more product is made faster. We continue. The use of a continuous process helps to make method 2 cheaper. Suggest why? Well, this process can be automated. So needs fewer workers. The implication there is that they therefore have a smaller wage bill. And then it says, using the table, explain one other reason why method 2 is cheaper. Well, here we note two differences in the table in the last two parts. We might say, for one other reason, it gives a higher percentage yield. Or we might have said that because there's no waste products made, there's no need for pollution control or in order to get rid of those additional products which are unwanted. So here's the answer. Catalyst makes the reaction faster, can make more in a shorter time. Advantage of the continuous process is it can be automated, and no need to pay for pollution control or higher percentage yield or various other possible answers, as you can see. And now we look at why it's often expensive to make and bring new pharmaceutical drugs to market and why it's difficult to test and develop these. Let's look at these reasons in turn. The first is that it takes a very long time to develop a new drug. For example, ibuprofen, which was patented in 1961 as Nurofen by Boots in Nottingham, took 8 years in order to come to market because in that time it had to be manufactured as cheaply as possible, had to be tested first on animals, then on humans in small and then large scale tests, and all of these things have to be done in order to satisfy government legislation to make sure it's safe to release to the public. Secondly, research and testing is very labour intensive and expensive, and it requires highly skilled and highly paid workers in order to do this. Raw materials can also be very expensive. Some raw materials for new drugs come from, for example, South America, and therefore they have to be extracted and transported to Britain in order for them to develop the drug before perhaps those raw materials can be made synthetically. Labour costs are always expensive because the people who are developing these new drugs are highly educated and therefore highly paid. Energy costs are always expensive in industrial processes, for example to maintain high pressures and high temperatures. And marketing costs are very, very high for pharmaceutical drugs. Pharmaceuticals have to be patented or copyrighted, and this copyright only lasts for a limited number of years, so the company needs to make as much money out of the drug as they can in the time when they are the only people allowed to make it. If it has to market this new drug aggressively, that's going to cost a lot of money to market it to doctors and also to the general public if it's not a prescription drug. Here's a past paper question. A new anti-cancer drug is made from a rare plant only found in South America. Less than 100 kilograms of the drug is made each year. It's made in a batch process. The cost of manufacturing and developing this drug is very high. Write about some of the reasons why the cost is very high. Well, the raw material needs to be imported and will be expensive, and we'll say that research and development is time consuming and needs expensive workers. The anti-cancer drug is made in a batch process rather than a continuous one. Suggest one reason why. This is because demand is low, so it must be made when it is needed. As you can see, there are very, very many reasons why these drugs are expensive, and a lot of different reasons are acceptable, but you need to learn 3 or 4 of these in order to make sure that you've got enough information to answer a question such as this one. Here's another part question. It says it often takes years to develop drugs before they can be used by the public. The drugs need to be thoroughly tested and researched before use. Write about 3 other reasons why developing and making new drugs is extremely expensive. Well, we might say, for example, that raw materials are rare. We can't automate some processes, So it has a high wage bill, and finally there's government legislation to satisfy. And again, as you can see, there are plenty of other reasons that you can give, although it doesn't allow marketing costs, curiously, on this one. Next, we look at how the chemicals for making new drugs are extracted from plant sources, and there are 3 key terms you need to learn. As you can see from this table, many drugs are made from plant materials, and so the active ingredient has to be extracted from the plant material to be used as the raw material for making the drug. The first stage of this is crushing. This breaks up the cells in order to release the compound into the mixture. The useful compound has then to be extracted by dissolving in water or another suitable solvent like alcohol, followed by filtering off all the plant material. And then that active ingredient has to be separated from the other ingredients which are dissolved in the mixture by a method such as chromatography. Chromatography works by these different chemicals travelling at different rates with a solvent through either a powder or through paper. Here's a past paper question on this topic. Medicines and pharmaceutical drugs are specialty chemicals. Digitalis is a medicine used to treat heart disease. Digitalis can be extracted from the foxglove plant. Describe how chemicals such as digitalis can be extracted from plants. Well, first of all, the plant material is crushed. It then is dissolved in a solvent. And purified or separated by chromatography. So the 3 key things you need to remember are crushed, dissolved, chromatography. An alternative to chromatography as this separation method is distillation. Finally, how do chemists or pharmacists tell that a material is pure or not? Well, there's 2 methods of doing this. One is by using either its melting or boiling point, and the other is by running a chromatogram. And it's important that drugs are pure because the presence of any harmful impurities could make people ill or have side effects, so drugs have to be regularly tested for purity. Each pure substance has its own melting and boiling point, and these are easy to find in books or on the internet. If the substance does not have the same melting or boiling point as the published value, it means that there are impurities present, because impurities will raise the boiling point and lower the melting point. An alternative method to test for purity is to use chromatography. Each substance will have its own particular pattern of spots, and additional spots suggest impurities. So if M here was our pure medicine, we could see that samples 1 and 3, because they have the same pattern of spots, are the same substance and therefore are the pure drug, whereas sample 2 has this additional spot suggesting an impurity.